For our last talk of the morning, we're going to hear about methods for automatic annotation of the Maize genes. Josh Kangas is presenting along with Rebecca O'Connell, and they're joined in the audience with other team members, Chris Stubb and Krista Manton. Their faculty mentors are Dr. Brent Buckner, Dr. Diane Janet Buckner, and Dr. John Beck.
First, we'd like to thank you all for coming and do bear with us for this last presentation for lunch. All right, so in order to start the proposal, I'd like to give some biological background. Rebecca's going to help me with that. So we're going to talk about the true meristem and functional annotation. Then we're going to discuss the previous efforts in terms of the artificial neural networks, classification, vocabulary development, and assessing the accuracy of the system. And then we're going to talk about the current goals of the system.
Okay, so the sheet apical meristem, which is also known as the SAM, is a small group of undifferentiated cells that will provide, it's about the size of a grain of sugar, and it will give rise to all of the above ground plant tissue. It's located about right there on the 7-day seedling. This is essentially the same as stem cells in animals. And you can, on this, it's a longitudinal section of the SAM. You can see the SAM here in the middle, and then the newly differentiated leaves wrapping around it, the leaf primordia. In this scanning electron micrograph, you can also see the same thing, SAM, and then the leaf primordia wrap around it. For analysis, we need to extract the SAM so that we can get a pure sample, and we can use that for microarray analysis to determine which genes are differentially regulated. Laser capture microdissection is used, and it can excise a small amount of tissue and as a pure sample that can be used for the microarray analysis. The Maize genome has just recently been sequenced, but most of the accessions have not been annotated. And we have 4 different Maize chips that have approximately 40,000 spots on each of them, which covers over half of the genome. And the cDNAs, which are copies of mRNAs taken from the SAM, are added to these microRNA chips to be analyzed, and they show which genes are up or down regulated, differentially regulated in the SAM tissue. What our group is doing is we are attaching biological information to each of these accessions, which includes biological processes and functions. To do this, we have a set protocol that we use to look at each of the unique accessions. Each one takes between 20 minutes and 2 hours to complete, depending on how it is. We start by doing a BLAST-N to search the nucleotide database, and then a BLAST-X to search the protein database for sequence similarity. And then we find a longer Maize sequence, and we use that to go into this program FGeneSH, which gives the predicted gene and its corresponding amino acid sequence. The amino acid sequence is then used to do a BLAST-P and an interproscan. Interproscan looks at the different protein families and protein domains and compares to look for similarities that way. This is very helpful because it helps us determine a function. Interproscan also gives gene ontology or Go numbers, which are great because you can compare the gene, the identified gene, and its function across other annotated genomes. After the BLAST and interproscan, we usually have an identified gene name and a function if there's one available. So using that information, we can place them into categories. These categories have been developed based on other bioinformatics studies. And here is the view of the categories that we use for our study. They've been adapted from the other ones to fit the SAM characteristics. And this is the view from our database. In addition to the gene name and the functional category, we also do literature searches to find relevant literature based on the identified Maize gene or its homologue in another organism. This is very useful to future studies. We've had the NSF grant for approximately 4 years, and we've annotated 6,600 of 40,000 unique accessions with a team of 15 different students. This basically means that 15 Truman State University students have annotated over 1/10 and produced value-adding annotations for over 1/10 of the genome. And this is really significant and we're very excited, but it would be great if we could automate certain parts of this process to make it go a little faster and expedite future annotations.
So next I'm going to talk about the previous efforts of this project. So we wanted to develop tools to automatically annotate these genes with 90% accuracy. So what we could do with this, as Rebecca stated, we could use this to expedite the process of annotations. We can also use it to check annotations that are already done. And we can facilitate further research in metabolic and genetic pathways. We also, as a last one, wanted to store and display the results in a meaningful way so that it can be published. So the question we have here is, how do we find the function of this gene? First, we need to translate from nucleotides to amino acid sequences, and then we compare this amino acid sequence with all known amino acid sequences. And prior research indicates that if you can show that the sequences are similar, you can also reasonably assume that the functions are going to be similar. So the approach we wanted to take was artificial neural network. So an artificial neural network pictured here is a mathematical representation of a network of rudimentary neurons. And you have nodes here and connections here. Each of the connections has a weight, and each of the nodes has an activation value. So why did we choose an artificial neural network? We chose an artificial neural network because it was well suited for classification problems, which is exactly what we're doing here. They have the ability to operate within a certain level of noise, so they can ignore spurious data. They also have the ability to learn patterns by usually recognizable likeness. So in the last neural network, I had 2 input neurons and 2 output neurons, which I'll explain more. But in the neural network we're using, we have approximately 500 input neurons and 1 output neuron. So for a human to learn the patterns of 500 inputs would be very difficult. It's also important to note that artificial neural networks are essentially a black box. So you have inputs, you have outputs, or you have inputs there, outputs here, and everything in the middle is really difficult to pull any kind of meaning out of that. But it's still worth explaining so that you can understand how the neural network works. So that would be the organization of the network. So I'm going to tell you about training a neural network based on the exclusive OR function. We use what's called a backpropagation neural network, which means we have inputs, and then we set the inputs to known values such as these, and then we set the output that, and then we activate through the network based on these inputs. We also, in our network, have what's called a bias node, which is a neuron that is always activated. And the reason for that would be, in this case, we have 2 zeros. Based on the activation function, we would end up with no output here if we had no bias node. So the training process starts with a neural network that has random weights between all of the nodes. And we start with the outputs at 0, 0. Set the inputs, and then transmit the activation energy. So if we would like to calculate the activation here, the next step, we take this value times this value, plus this value times this value, plus this value, plus this value. And the bias is always activated, so it's 1 for the energy. And then we take the sigmoid function of that sum. And then we continue the process on through the neural network, and then we end up with an output. And based on our inputs, the output should have been 0, which is 0.72, so it's way out, that's where it's at. So we need to go back through the network and correct for this error. So the error is propagated back to the network. And you can see the weights change corresponding to what needs to be done to reduce that final output in this case. This process continues on. And then we repeat with different inputs. So we change the inputs, and the weights are going to change also. In the end, we have a trained neural network. So these are the actual weights that would come from this particular neural network and the implementation that we use. So as you can see, the inputs here, the outputs, and the correct values, the outputs from the neural network are relatively close to the correct values. This is after 1,000 of these training cycles. 1,000 is quite reasonable for this particular network because it's small, but the neural networks we're dealing with are large, 500 inputs. The training process takes a really long time. So we use the XGrid server. So class X. The question is, how do we apply the neural network to this biological problem? We get our information from class X. So this would be the results from class X, which is the process I explained earlier. We have p-values down this side, which is basically a measurement of the closeness of the hit. And over here we have links. And under some of these links are PubMed abstracts, basically. So this would be an example abstract. So a biologist could read through this and fill out some sort of meaning about this particular paper. So what we're going to use for our inputs would be the presence or absence of specific vocabulary words in the abstracts of the related results from the class. In the output, we have, for each of our categories, we have a neural network that outputs from -1 to 1, with low values representing lack of membership and high values representing membership in that particular category. So this would be, okay, so I was afraid this was going to happen. I think, so I made my PowerPoint presentation. See, I don't think my position everything's going to look that well. So these should be over cellulose, elongation. That one was over pectin, and that was over membrane. So I was trying to indicate the specific words that are useful in this. This would be a short example vocabulary and what the inputs would look like based on the abstract. You can notice cytoskeleton, that's shortened because we could have cytoskeletal, cytoskeleton, etc. So then this would be another shortened version of the neural network. So you see, you have the inputs, which represent vocabulary words, and they're either 0 or 1, and then you have one output. Our neural networks we use extend 500 words, and the hidden layer here extends 30 to 40, depending on which particular neural network you use. So this would be further illustration of how the neural network works. Up here we have the vocabulary words. We have accessions going down next to it, 500 vocabulary words, we have 40,000 accessions, and this is just for the cytoskeletal neural network. So the manual annotation of this particular accession would be cytoskeletal and transcription for this one. The expected output based on this, because it's the cytoskeletal neural network, is 1 and 0 is 50. So we can look at the outputs here and try to make some determination of the annotation. Highlighted these parts because this shows the strength of the neural network. These words probably have something to do with cytoskeleton, so you can tell that the neural network has learned the pattern of cytokinesis being present, that probably indicates cytoskeleton vocabulary. Whereas the opposite is true here. The cell wall doesn't seem to have any pattern, so that's recognized as noise and basically ignored in the neural network. The vocabulary development is a really interesting process as part of this project because this was where we really were able to collaborate with the biologists. And also, this is the unique part of our project. We designed the vocabulary so it's specifically meant for the Truman State of Maize and to pull out the functional categories of that particular part of it. So we started out with the basic vocabulary, and for each of the words, we asked the biologist, does this word in an abstract indicate to you a specific function category or a small group of specific functional categories? If it did, we kept the word. If not, we removed the word. If it meant too many, or if it would indicate too many categories, that would be noise for the neural network. If it was too few, that could give spurious results in the training process. So beyond that, when we looked at words, when we kept them in our list, we checked how many times they showed up in abstracts. If they showed up a few times, we removed it. If it showed up way too many times, we removed it. And then after that, we would test each category for its accuracy. And if we determined that the category needed accuracy, we would try to pull out keywords, or try to add keywords that would indicate that specific category. And every time we made a new vocabulary, we had to train all of our neural networks and then retest them. So after 8 iterations of this, we have approximately 500 vocabularies. The next important part of this would be assessing the accuracy. For a neural network training process, you need 3 different sets of data. You need the training set, which is what is fed into the neural network You have the validation set, which is, you don't expose those to the neural network until the testing process is within the training process. So you want to make sure that your neural network hasn't overfit your training data. You check to make sure that it's still improving in accuracy on the validation set. And then finally, after that, you have the testing set. So once training is complete, you test based on that set. So this would be the abbreviated software output. So we have 22 categories, 40,000 sessions. And we have all these outputs. So we need to determine what are each of these neural networks saying about these sessions. So the first method we used was to take all of the outputs for one particular session and rank them. So if we look at the top hit in the 13 of the 21 categories, was statistically better than just guessing that answer. And I say 21 categories here because of the restoration of data for this test. We go all the way down to the top 5 hits, 19 of the 21 categories were better than that. So the next method we could use would basically be a cutoff method. So we use activations when they were in the range from -1 to 1. So for this particular illustration, we use a cutoff of 0.7. So this one definitely belongs to metabolism, this one definitely belongs to transport. With a cutoff of 0.7, however, we could potentially miss annotations like this. So it might be useful to have a person sit down and look at this particular session and see why it's so strong in transport. We also need to minimize false positives. That's the same situation. Using a cutoff, we use McNamara's homogeneity test, and with a cutoff of 0.5, 8 of the 16 categories were homogeneous with the manual annotation. And at 16, again, because I used for this physical test, we didn't have enough inputs in some of the categories. And at 0.3, we have 15. So the next sort of analysis we wanted to do was a logistical analysis. Because we're using sigmoid function, we were looking for a certain cutoff where we could minimize false positives. And what we found is that across different categories, the cutoff required to get 90% accuracy was different. As you can see. So now I would like to discuss what we're currently working on. So as a result of being, this is the second year of this project, and this is a really results-oriented program. So our number one priority is to prepare this manuscript for publication. Obviously, this is the template. There's definitely meaningful content in this, and we plan on submitting this for publication by the end of the summer. Partly because I'm also going to be gone. So in trying to determine what we needed for this manuscript, we realized that we need some sort of data to validate our method. So we're doing what we call the bucket test. And the bucket test is a really common test that you can use for neural networks. If you're familiar with phylogenetics and bootstrapping, it's similar to bootstrapping. And the reason for that is we take all of our data, our known inputs and known outputs, and we split it up into 4 parts. And then we randomly permute those 4 parts. So 2 of them go to training, 1 to testing, 1 to validation. And then we completely train this neural network configuration and get the testing data from that. So then we do that 12 times. We only do that 12 times because of the time it would take to train all of these neural networks. So one of these tests takes about 2 days. And we got 12 of them. And that's just for one single category with one configuration of the neural network. So that's why the distributed solution on the Mac cluster really helps, but also limits what we can do for testing. So then the next test we wanted to do was a human test. So to show that our method is actually working, we wanted to take human genome, human genes with known Go functional categories and use the same method. So we'll pull out the vocabulary information for the last result of the human gene and then compare the results to the Go categories. And what this can show us that it can be applied to other well-annotated organisms, and also shows the strength of the well-tuned vocabulary. So we expect that using our vocabulary that's been trained for the true data comparison of Maize, we expect that our accuracy will not be quite as good for the human. The second test we'd like to do would be the Arabidopsis test. So we want to show that two organisms that are relatively similar, we could use one neural network to annotate the other one because they're similar. So we'll use the neural network trained on the vocabulary that was built for Maize to annotate the rapid genes. And because that's a well-annotated genome, we can test our results. We could also do this with the human-trained annotation system on mouse genes, because those are also well. So the final goal we have, we did not finish the public access website for last summer, so we're going to continue to work on that. And what we'd like to do with this is display neural network outputs and allow users to enter sequences to be annotated using our Maize trained system and allow for higher results. So we kind of have a beta version of this website. So we have all the categories up here and sessions down here. And we tried to color code based on the activation. So this particular area was interesting, so I'm going to use that to show what we'd like to do with our cluster. So this would be completely unordered data. And at this point, you can tell by the category and by the session, you can see interesting things, but you can't see relationships between these. So if we cluster based on the accessions, we get similar accessions grouped together. And what we'd like to see then after that is we cluster our categories. So particularly these two accessions, they hit strongly with transport and with metabolism. So we might like to have a biologist sit down and determine what makes these two useful for transport and metabolism. Well, there was the team. They're very good, obviously. I'd like to thank Dr. Buckner and Dr. Buckner, Dr. John that's not here, and
We have time for some questions.
So your neural network did that hidden layer of those nodes. Is there always only one hidden layer, and how many nodes are in that layer?
so that's one of the problems that we had last summer. The hidden layer can vary so you can have more than one hidden layer. Research has shown by other people that having more than one hidden layer doesn't necessarily give you more resolution. Having at least one, though, is important. As far as the number of nodes in that particular layer, when we start out with a smaller vocabulary, we found that the accuracy was the same as we reduced. See, that's probably better. Basically, the number of nodes in that area. The more inputs you have, most likely the more hidden nodes you have. And that was one of the tests we had to do last summer. We had to figure out where the best number of hidden nodes. So some of them had as many as 40, and that really increased the complexity of the computation functions that added a lot to it.
The original weights that you have from the input to the hidden layer, are those just random?
Yes.
You're talking about turning down your vocabulary, being more specific. Like when you first sat down with biologists and built a vocabulary, how big was it before you turned it down?
It was kind of a trim and grow process. So the first vocabulary, I think, in the 80 words, and then 7. But in the end, it's just growing.
Josh, in your proposal, if I remember right, there you were talking about the fact that, at least for the first part of this project, you were focusing specifically on genes that were differentially expressed in the SAM versus in the elite primordia, for example. Just not knowing that much about the Schuettinger meristem, can you tell us, you said it's a fairly small cluster of cells. Is it small enough that the levels of gene expression are homogeneous throughout the SAM, or might it be even different in different parts of the SAM?
There's about 250 cells that make up the Maize SAM, and I'm pretty sure those have very similar expression enough that it's close enough that it doesn't make a huge difference.
Okay.
It's just once they start differentiating that you.
Then,
Yeah.
Hey Josh, Those numbers that you get, are they just useful for deciding plus or minus if they should fit, or is that a probability function? If this is 99% accurate or 15% accurate?
If you look at one particular number, it's useful for a plus minus. Whenever we're looking at one category across the board, we can use the statistical analysis to give a probability to that